Welcome to Careers in Discovery, your window into the world of biotech leaders. On each episode, we interview the industry's leading executives to understand how they built their careers, what they've learned along the way, and the advice they'd give to their younger selves. This week, I'm joined by Hazel Jones, CEO of Enhanc3d Genomics. Hazel shares her journey from her early days in biotech to leading a company at the cutting edge of 3D genomics. We explored Hazel's career path, her transition into a CEO role, and her insights on leadership, the significance of networking, and balancing professional and personal life. So I'm back on Careers in Discovery with Hazel Jones of Enhanc3d Genomics. Hazel, welcome to the show.
It's good to be here.
Fantastic to see you. We always start with a little bit about what you're up to now. And obviously, you've recently stepped into the role of CEO at Enhanc3d Genomics. Mm-hmm. Really interested to talk a bit about the company, first of all, and what you guys are up to, where you're at. And yeah, tell us a bit about what you do.
Okay, yes, so I'm CEO of Enhanc3d Genomics. So it's fundamentally a platform company, but actually it's the 3D genomics we're interested in and the data around that. And just to explain that, so the 3D genomics is the interactions between the enhancer regions in the non-coding genome and the promoter on the genes. And put more simply, it's if you can imagine your DNA is a ball of spaghetti in the nucleus and there's lots of touch points. It's those touch points that really drive the function of the cell. So all your cells in your body have the same genome, but your immune cell has a different 3D genome to your cardiac cell or your brain cell. So it does a different function. So it's really exciting to understand the non-coding genome because that's actually 98% of the DNA of the genome. Mm-hmm. And obviously in the last 20 years, we've seen, we've gone from discovery of the first human genome that cost, $1 billion and took several years to the fact now that we can use, it as a diagnostic test in the hospitals and on clinical trials. So we're really unlocking that additional data and enabling its use actually both in drug discovery, so we can identify novel targets in different diseases, so we're disease agnostic, but we can also use it for patient stratification to a finer level than some of the information that's used at the moment. So we spent the last 2 years really getting the platform right and the tech and being able to do the bioinformatic analysis of the data that's coming off. And now we're in that exciting bit of applying it to the different biology questions and working in collaborations both with academic and big pharma to show the power of that. And actually, we've had some exciting data this week to show we've really accelerated doing that target identification, in a certain disease area. And we've worked with a collaborator that's sort of taken 2 years to get to 10 reasonably novel targets that they're interested in. And with our technology, we got to a similar number, but in 3 weeks. So the potential of using this data and integrating it, obviously, with a lot of the buzzwords like AI, really is going to unlock that power of the genome sequence.
Yes, I see. And as you say, there's been much more work done on the non-coding genome over the last several years, and I guess a group of companies that are really focused on understanding it. How much of it do we understand at the moment? How far along are we?
So some bits really in depth. So there is mechanisms where if you're looking at one gene, you can do the fine mapping down. And typically, you probably get between 5 and 20 enhancers. So for every of the 22,000 genes in your genome, they all have those. And our technology allows you to see all of that. So we're really going after that data side, so we can understand it, but it can already be used. And actually some of our previous data hit the news a few weeks ago in, I think it was macrophages, because they've been able to do one of these distal regulated SNPs, had then to a target gene, and therefore you understand the disease mechanism, which helps, like we said, with stratifying patients or potentially looking for novel drugs. So it's getting out there, but it's the totality of what we can do, I think, that's really amazing.
Yeah, and just the understanding that there are
other causes or other factors that identify these diseases or identify these characteristics of patients, that kind of thing, that can give you different angles to find targets is
and the thing that fascinates me is again how many differences you can see in that 3D
okay.
profile. So we can tell the differences between T cells and B cells in the immune system, which again you can by a lot of different sort of biological ways, but we can see the difference between a T cell and an activated T cell. And actually, then we can look at those profiles in the disease. So at the moment, we're looking at lupus and multiple sclerosis, but you can start to really differentiate even a big class of diseases by the different profiles that you find in a patient sample.
Yeah, I see. Okay, interesting.
Mm.
And then in terms of where you're spending your time, Hazel, you obviously joined the company as COO, I guess coming up to a year ago now. Recently made the transition into the chief exec role. Has it been very different? How have you found that move?
I guess it's not so different. I was actually only 6 weeks in the COO role before
Okay.
I became interim CEO, and then more recently I've been given the position permanently. I think it sort of tracks back to almost that career. So I started my career in biotech in Cambridge antibody technology. Have been in and out of different organisations, but the majority of it's been spent in big pharma, AstraZeneca. And it was just wanting to be a bit closer to that science, a bit closer to see the decisions. So I knew I wanted to come back into biotech and took some time last year to think about it. My recent roles have been more of those operational ones. I'm very organised. I'm a planner, which is very useful in business. And so the COO was an obvious choice for me to sort of go into. And I think after a couple of weeks here, it was clear that the platform has had that technology developed. It's very, very strong. The IP is good. We've got a second generation platform coming out. We had a lot of use cases in the biology sort of arena, but actually getting those to be robust and be interested from big pharma was the step that's needed. So that's really where we focused for the last 6 months, and changing our sort of narrative to being a data company that can really accelerate drug development, both in that early stage with the targets, but also in the biomarkers, as opposed to a platform company that's really cracking that code in the non-coding genome. So the application's the same, but it's a different narrative around it.
Right, I see. Okay, so exciting times now. You've focused down, and
definitely, yes.
Excellent. Well, look, I'd really like to talk about your career as a whole and your path through it. As you say, you started out as a scientist, you moved into more operational roles, biotech, pharma, lots of variety in there. So I'm sure we'll get into all of that.
And it's nice in the fact, I guess if you go to LinkedIn, it looks like a really nice, smooth path. And I've done all this amazing planning, and sometimes it's not quite like that. So I'll give you a few of the quirks along the way as well.
Fantastic. I look forward to those. But take us back to the beginning, first of all. So you trained as a biochemist originally, but why science?
Well, why science? I think it's one of those things. It comes from, I don't know what comes first, the passion or the the being good at it. So it's definitely driven from those school years that it's something that I was very interested in, and therefore you spend more time in it, and therefore you're particularly good at it. Did look at medicine, and my parents, first generation, they were, you've got to be a doctor, you've got to go and do medicine. And I spent a bit of time with a GP, and I spent a bit of time in surgery where I nearly fainted and went, I'm not sure the medicine's the right way for me. And then, did that digging on my own to find pharmacology courses. So my undergrad was pharmacology.
Right.
Did a project in actually cancer research as part of that, and decided to do a PhD then in biochemistry. So very much focused around a drug. It was doing that sort of investigation. It was a drug that was very active, and we didn't really understand the mechanism. So that mechanistic piece of understanding disease and placing the drug in the right places has always interested me. And then moving to London, I actually got my first job by writing on spec. And again, it looks like it's a nice transition, but the reason was my now husband had proposed and moved the night before he moved to London.
Ah.
So it was it was not driven by total choice, but I managed to end up in a very small biotech called Zenova that was looking at multi-drug resistance, and had a great 2 years there, doing that transition from academia into, okay, what is a drug project and how do they run?
Yeah. And what were your early memories of that transition into biotech and into industry?
For me, it was actually quite smooth. We were actually in a university setting. We were actually on Brunel and head office, so where the labs were. We were a small team, so there was probably only about 10 of us working on that real biology mechanisms piece. A little bit of a shocker on their sort of 9 to 5, and actually someone at the end of the week wants to report into a project team of what you've actually done.
Right, yeah.
But for me, yes, a great start and understanding, more the drug development and what's required, as opposed to being an academic where you can, follow your nose on what you're interested in.
Yeah, of course. And then that eventually led you to a position at Cambridge Antibody Technology, as you said. We've had a lot of CAT alumni on the show.
There's a lot of us around, and it's a really, really close network in Cambridge, so that's great. And actually, I was telling someone else the story, so it was great. I moved, I think it was in 2002, and when I went around sort of my various peers and mentors, most of the information I had back was, no, that's a really ridiculous move. Antibodies are never going to be drugs.
Right. Yeah. Okay.
So I went with some trepidation in the fact it was a great position. I was taking a step up quite, early in the career to build a team around oncology, biology, and those mechanisms. And, everything I saw from the interview and everything, and heard, it was very exciting. But, if you look back now, you can say, oh, that was amazing. It was slightly luck and a risk on my point of view, because me and my husband moved over to Cambridge. So he gave up his postdoc and then was made redundant 2 days before he started his new job. So,
right.
Like I say, it looks smooth, but there's all these bumps that happen along the way. And I still remember it very fondly. I'm sure if you've had alumni on here, that experience was amazing, both on the pace, but also just being very integral to actually what they were trying to deliver. It was I think around 150 when I joined.
Okay.
Very tight, they had a lot more in respiratory and that sort of thing, but it was great to be part of that journey.
Yeah, absolutely. And I'm fascinated by this because that company has had such an impact on the sort of UK biotech ecosystem, both in terms of what they actually did, but also then what the people who work there have gone on to do. And I'm just fascinated by what you saw there that you think, what do you think made it as successful as it was? What were the things that stood out to you?
There's two sides. I think there's the culture and being part of it. And I wasn't very senior at the time, but I felt I was updated, and it's been interesting since I now know Sean Grady and AZ, and I've heard some of the stories, as it went along, and what AZ was thinking from the other side. But being in CAT was it was like being part of a big family.
Yes.
And they were training you up as that sort of scientist and person. There was a lot of investment in you personally in that respect. And the systems that were around the science was driven by passion and excitement, as opposed to like a real delivery piece. And I think, I don't know why it's so sad. I don't know whether part of that training then enables you to grow. I really feel I grew up there as a scientist. My scientific training was there, and they made a lot of big, bold decisions when they were still actually quite small. So that belief in, if you know the science and you believe that's the way you should go, I think has stayed with a lot of us. And that's why I can now count myself in one of those, the number of CEOs around Cambridge that come from that team, and it's impressive.
Yeah, because a lot of companies today even, talk about being led by science and led by data, and, some of them are more committed to that than others, right? But they were at CAT, you feel?
Yeah, definitely. And the discussion was had. So, you have the exciting discussions, but also the very difficult discussions. So, it was it was everything you got exposed to. You weren't, in the lab doing something and someone else was making this decision. So, but I think that's, I don't know, that's true of a lot of biotech, but.
Yeah, I think so. I think it's difficult to balance those things, isn't it? And particularly as you grow, if you go quickly, keeping sort of your hand on the wheel and steering in the right direction can be a challenge.
But I remember fondly, it was that I hated them to start with. We had Friday morning lab talks, which was basically the whole company in the room. That obviously clearly got bigger over years.
Yeah.
But it was that opportunity to present what you were doing to everyone. But it felt collaborative. And it was it was right. I can go there first and say something before going, to a big governance meeting or maybe going out externally. And I don't know whether you've spoken to John Elvin. He always had the questions, and he always, it didn't matter whether it was a quite simple question or very difficult. He'd just want to ask it, and that encouraging that audience participation. And I'm asking it because I'm interested in it, not because I want to put you in a difficult spot. So it does come back to culture, which again is discussed a lot now, and obviously I want to recreate some of that. I don't know what the magic sauce is, though, unfortunately.
Fair enough. But you were there for a good chunk of your early career, about 10 years all in, with the transition into MedImmune and everything that happened there. Did it change a lot when it became part of MedImmune, do you think?
It changed as it grew. Partly that was numbers, partly because obviously we're trying to work collaboratively with teams that, were across the pond. It was different. I mean, again, and I was getting older and taking on more responsibility. So then it was, looking at the projects, ensuring that we had the right balance, doing budgets, talking to our counterparts that were, managing similar sized portfolios. Some of it worked really well and was really exciting. And some politics got in the way on occasions, which is sort of anything of that size. But I think they would like to think there was give and take from both sides. They started doing much more open offices, which obviously builds that collaboration. And eventually we got into that working relationship and had an amazing portfolio of both small molecules and antibodies. And that's quite an accomplishment. So yeah, it's nice. It's just nice to go in. I was lucky because I didn't change site. I changed buildings quite a lot on Park, but I got to experience all of that in different ways. So, and I think because there were multiple connections, both in the first case with AZ and then with MedImmune, you learned that some of them were very easy and very good, and I'm still in touch with some of those people. And then some of them, be it because of the science was difficult or the project wasn't getting the support because we're not being driven by the science, then it was more difficult.
Yeah, I can see that. And then 10 years is a good stint anywhere. And I think it's typically a period at which you sort of start to assess where you're at and think about what options are and things like that. And you then went and joined what on the surface of it would look like a very different type of organization in cancer research. Tell us a bit about that move.
So I sort of did a sidestep more. So cancer research, the CRT, the technology drug discovery bit, was more like the original CAT. So it was a small organisation focused on drug discovery. A lot of it building alliances with biotech or pharma. They really used my collaborative skills in that piece of being able to go out, talk to pharma, and then go out and talk to an academic and somehow get them to agree in the middle. It was also interesting for me of taking a step up and so going a little bit out of my comfort zone of, including target ID and validation and everything like that. And so it's that sort of wider remit and going from large molecules, antibodies back to small molecules. So, and the drug discovery pipeline is the same. And now lots of people do the large molecule, small molecule mix. But at the time, it was quite different. And we did manage actually to bring some biologicals into into CRT. And then I had a great opportunity sort of after doing that for 2 years. The Combinations Alliance was part of the CRUK sort of head office, one of their big sort of collaborative projects with industry, but looking at phase 1 trials. And so I was able to step up on that, again, bringing that collaborative style that I'm very used to, of going, right, we've got one person over here that needs this, and we've got the 17 ECMCs, which is the Experimental Cancer Medicine Centres, so all the phase 1 units that want to deliver some trials with this person, and sort of matching them up. But with that, the science was relatively okay, but understanding, setting up a phase 1 study and everything that comes with it was, again, a massive growth and learning phase for myself, which puts you in a really good position as a drug discovery, drug development, when you've been able to do a little bit in both camps.
Yeah, absolutely. That's one of the things I was going to ask, actually, was that kind of your first real exposure to clinical research?
Yes, I've done a little bit in MedImmune in the facts and you know, preparing those INDs, but it was very much, and they don't do it so much now, but you sort of go from discovery into development, and it was almost like throw, and then depends whether they want catch. Actually, now in most organisation, that's a continuum rather than a one organisation, part of an organisation handing off. To have that experience, and also just what I was exposed to, because actually the CR UK Drug Development Office did a lot of phase 1 trials with pharma themselves. So I was, you know, doing the partnership piece, but I understood a lot more of, you know, the data management, the setting up, the what is needed very operationally, as opposed to, oh, you know, this is a really exciting drug, we should put it into patients, and it's got to be safe, the tox package has got to be, you know. So I think most of my changes have been to enable learning from my side, which gives me that wider perspective.
Yeah, absolutely. And a lot of the people that listen to this podcast are, you know, drug discovery scientists, or they're leading drug discovery programs or that kind of thing. What do you think would be useful for them to know about clinical studies that might help them to do the right kind of research, to develop the right kind of data, that kind of thing?
I think you can't beat that, just the experience of being in there doing that. So although, you know, I wasn't doing the setup, I was managing people that were sort of doing the operational piece. So you have to understand all the documentation and everything like that. And you've really got to get in the weeds to have that appreciation. I've also I also did a secondment for a year at AstraZeneca into digital health, and that was similar thing. So it was looking at using all the data that's out there to increase numbers into recruited for clinical trials, and that can be anything from the electronic health records that are very different in the NHS to they are in Europe, to obviously, and some of the data from companies like ourselves, which can be very useful for pulling people in. And you can identify them, but the link between actually identifying them, getting them to a centre that's running a trial that's, you know, really useful to them, and actually then managing to bring them into the trial is very difficult. And you know, you get more appreciation, but I think it's another thing that you have to go down into the weeds and do one slice of it so you can see how it matches with it. You know, you don't have to spend your 20 years in it to understand, but I did a year and I came out so much more knowledgeable of just how difficult clinical trial recruitment is, and the numbers are incredibly low. So I think it's down below 10%, apart from oncology in the UK, which is really driven by CR UK being such a good advocate of doing those. And as we move forward, you know, we want those numbers to increase, because, you know, the way to get best molecules through is you need the people in the trials. But for people in the careers, I think it's having that varied approach, you know, either to different disease areas or, you know, very early in target ID and then some clinic. And it's hard to know where they're going to come from. You know, I didn't plan. I knew that I wanted at some point to put clinical in my, you know, in my CV, so that I'd have that experience. And I think it's looking for those opportunities, talking to people in those areas, because, you know, there are usually projects where you can, you know, make a difference and learn something new.
Yeah, absolutely. And that sort of seeking of new experiences, do you think that's something that came naturally to you? Did you have to push yourself to do it? What is that part of your makeup, do you think?
I think it's a little part of my makeup, and I think I got good feedback from it in the early career. In the fact, you know, when you push yourself and do something different, and then it's like, oh, now someone's come back to me and it's following on. I think there's a diagram where you have your comfort zone. Can't remember what the second, then you go into your panic. And the more you push out of it, the more likely are. But it's once you've done it and you haven't had any negative effects, or the negative effects aren't massive, which is what you sort of think into your imposter syndrome. I couldn't possibly do that because I'm going to drop all the balls. That then you're encouraged to do it again and again. I think there's a little bit of determination and wanting something, but there's another thing of actually, when I've been throwing myself in the deep end, okay, I just need to keep reading to learn about this and it will eventually feel better.
Yes. Yeah. We often see this with people when they move countries or move locations. You know, the first move is the big one psychologically, even if it's not very far. It's just, no, I couldn't possibly do that because, and then as soon as you've made that one move, all the other moves become so much easier because you realize actually it's probably going to be okay.
Yes, and I think there's also, so I have 3 kids as well, so there's always, you know, like how it will affect everyone else, and it's just like, okay, I can do it here and now I can't do it over here. So that was another reason to have 6 months off. Yesterday I had Josh doing A levels and Luke and Rhiannon doing GCSEs, and I just couldn't pile on top of that my level of stress at work. It's just like this household is not going to survive.
Yeah, I can see that.
Choosing the timing is also, but and then taking the opportunity when it comes. You know, I did come into this wanting to learn about biotech again, so that I could take a CEO role, and now I'm doing it and learning as I go, and I've got, you know, some great support around me, and that's important too.
Yeah, absolutely. And you mentioned this at the top of the conversation, that sort of getting back to biotech was one of the key drivers for you, because you spent time directly before this at AstraZeneca, of course, which is very much the other end of the scale. So tell us about that part of your career.
So I've and I think you speak to anyone from CAT, they will hark back to those days, and what we actually have as it's like, is it just because, you know, you are going down the pub after work, you know, that part of it, that's where we felt we did some of the growing up, or was it truly amazing? And then, you know, I've had several different roles in AstraZeneca, you know, that I say, you know, I've enjoyed thoroughly, and then there's always some politics in there, because that's just big, you know, a big organization, you find it in any industry. But I think it was coming back to something and really shaping something from the start, and being able to, you know, I spent a lot of my career planning where that next stage is, so I learn all these skills. And now I need to apply all the skills. And although you can do that in big pharma, you're turning a tank around. And there's lots of other people that, you know, have incredible expertise. Whereas, you know, coming into a small organization like this, I have some unique expertise. I have another, you know, couple of members of SLT that are very different to me. So we make a good team. And then we've got some incredible scientists, you know, both on the biology side and the bioinformatics, and actually driving that from a platform and some exciting science like the 3D genome to, we want to change healthcare. You know, that's a 10-year window in doing that, and really maximizing the potential out of that by collaborations, which I you know, absolutely love driving forward.
Yeah, absolutely. So you found yourself in a place, as you say, perhaps not entirely by design, but that kind of brings all of those things together. And I think this point around, we obviously talked about the sort of potential in drug discovery of the sort of work that you're doing, but I think this point around patient stratification is really important as well, because yes, we want to drive enrollment into clinical trials, but it's also about enrolling the right patients, right? And you know, we're seeing much more that early stage research companies, biotech companies, are particularly using bioinformatics to analyze clinical data to sort of start to drive that end-to-end understanding. But this could be, you know, if people can get this right, this could be a real shift in success rates, a real shift in cost of drugs. You know, there's a huge amount of impact that could come on the back of it, right?
Yes, and it's trying to make sure we're following all that potential. And it is difficult. So, you know, you look at the different AI models in drug discovery and how the information is used. It's what is that critical element. There's a piece that sort of needs solving, which is sharing the data. So we can really take advantage of whole genome sequencing, but it is in hospitals, it is with academics, it is with, we're doing different collaborations to access that and really drive it forward to show the potential. And then it is making sure that we're connecting with the right people that can make it happen. And so some of the biotechs are really investing in drug discovery and new modalities, which is absolutely fantastic, what we need for patients. What we're going down more is creating that database, so we call it our 3D genome atlas, that we can then maximize what's happening from it. So we can work with the pharma partners for identifying novel targets. We can work with the diagnostic companies for trying to pull that information apart, at the same time as taking the technology forward to make it easier and faster to do this sort of analysis. So, it goes back to the genome's been around for 20 years, but, you can now go to companies and get your own genome done. What does it tell you? Well, it'll give you some risk factors for disease. The work we've done internally now using that non-coding bit and the 3D genome is that we can improve the accuracy of those. Because we're using that non-coding genome as well as the genes, but we need to do that across all the different disease areas. We're only a little company at the moment, so we've got to get this next bit really right as we scale and work with the appropriate people. That means then, the whole community can benefit, as well as everything else that's happening, alongside.
Yeah, absolutely. And as you say, proving the approach and then expanding it is important, right? And there's so much you could do.
Very much so.
Focus.
Yes, that's why my book keeps saying focus. What are we actually going to deliver in the next sort of 6 months?
Yeah, absolutely. I hear that a lot around the industry.
But it is, and I think typical scientists, we are interested and passionate, and it's you want to change the world,
right?
But you have to do it sort of one patient and one disease at a time.
And this is a perfect bridge to my next question. Thank you very much. It's like I gave you them in advance. But that sounds like maybe one of the lessons that you've learned over the years is around sort of, not tempering that vision necessarily, but bringing it to a practical place where what's the next step is kind of the key bit.
Yes. Definitely. So it is, you can have those big strategic plans and dreams, but the only way you're going to get there is actually what is the first step. So actually having that conversation, and then it's all delivery. It's really, really difficult, particularly when we're talking to collaborators at the moment, because we're like, we've got this big 3-year plan and we can get to know this and this. And it's like, okay, now step backwards. What is the first, sample or bioinformatics analysis that we're going to do that's going to give us something that you can make it? And it's the decision making. I think that comes a lot from the training in AZ, particularly as you see the big governance board going up, that actually if you're going to a phase 3 and someone's making £100 million decision, you've got to have good data that backs up that decision. And that sort of falls all the way back to even committing to a drug target in the first place. And it's already out there and being published how much genomic data underpins the success rate in drug discovery. I think it's at least twice as likely to have that. We think we can layer on top of that. AI has the ability to go out and find all that information and bring it, so you're not reliant on where I was a scientist and finding those papers by actually doing a search. AI will deliver them to your door every day, every week, or whatever you would like, and not just locally, on a global level. There's a lot more information coming out around the world now that you have to keep on top of.
Yeah, absolutely. And do you think that maybe, and I mean this in a positive way, sort of increases the onus on people who are running early stage companies to really understand that long term? Because you will know this as well as I do. There's lots of biotech companies do really good science and then realize there's not really a need for it way later than they should, right?
It's definitely having the discussion, so you know what you're after. And we're having, if we take that early stage at the moment, we're having that balance between identifying novel targets, which we know our technology can do. We've got evidence of that now. But from my time in pharma, everyone's doing that by different means. It's more defining the biological mechanism of a set of targets and prioritizing which one to take forward, because it's likely to be superior because of the mechanism, or there's you've already got that patient marker, so it's a simplified, hopefully faster route to clinic and eventually the patient. So we're learning all the time, and it's being disrupted all the time by technologies like ours and AI, but also by the change in the sort of patient pathway. So as new targeted drugs get to the market, then they get it different, as particularly in oncology. The ones that were in sort of stage 4 have come forward, and they're really changing early diagnosis. So it's a constantly changing field, and understanding that and sort of planning, going, okay, that's in phase 3, so in another maybe 2-3 years that disease is going to have a change in pathway.
Yeah, I see. Yeah, and I suppose there's an interesting balance there between being cognizant of the changes and taking them into account, and then understanding, well, what are the actual underlying principles and what are the things that don't. And obviously AstraZeneca have done a lot of work around this in particular, haven't they?
Mm-hmm. Yes, definitely. And they've all, they have been driven by the science, which has been fantastic, and to be a part of that. But actually it does throw up more challenges. So actually when you look at the portfolio, and again, even a big company has a set budget, you have to make very difficult decisions, because it's not like 3 of the projects have fallen over very early on, because actually you've got the information now that you've learned from those mistakes. Even with PK and tox and stuff, there's a lot of prediction we can do before even taking those molecules further. So it's interesting to know, to look at those success rate, do the analysis, but very cognizant of what's coming through the clinic now. So if we're thinking about targets, well, these are all going to be on in the patient pathway when we get to take ours through.
Yeah, absolutely. And then looking back at your career, Hazel, what would you point to as sort of the other key lessons that you've learned along the way that have really, really helped you progress, or maybe the things that you wish you'd known at the start, but
so I'm always good at volunteering for the not so glamorous jobs, because they get me into the meeting rooms of so understanding what's happening at the next level, and I can do it reasonably easy because I'm organized and just setting those up. So I've always, for my boss, tried to be that right hand person, so that I'm the one that's taken if something exciting is happening when you hear it earlier. So that's just the way I've done it. The one thing my husband will probably pull out is just I think he calls it my hyper focus.
Okay.
And that differentiate between projects. So really for the last 10 years I have to jump, 1 minute at AZ I'm doing COVID, and I was doing Brexit, very boring things, and then I'd be in a governance meeting or preparing the budget. So that ability to do that change very quickly from meeting to meeting and still keep spinning plates. I know my new CSO Dan says I'm doing it while on a bouncy castle, so everything's all a bit more wobbly. I think that hyper focus comes in on also that work-life balance.
Okay.
So I am prepared to work very hard, but I'm not really prepared to work at weekends unless I'm traveling for work. So it's a switch off and a start, and holidays. And when I'm present with the kids and doing that, I'm present. I took the time to have the time off because I couldn't do both.
Yes.
And I don't think anyone's got that bit solved. I think it's a changing piece.
Is there anything that helps you with that? Is there anything you've learned to do that? Because I think you're right. I think a lot of people struggle with that separation, particularly now there's so much hybrid and remote working. And
yes, I mean, I guess now with teenagers, it's that communication and a shared calendar. And
okay.
I did not lose my son. We weren't sure where he was. He turned out to be actually at the school open day, which he had told us about. And my very helpful son in Thailand said he hadn't seen him. So that connection is very important, and being able to do that in the right pieces. So I'm very happy, and was even in the early years to take the afternoon off because it's their nativity play. Because actually I've just worked till 10 o'clock 3 nights a week, and I had that's the bit they're going to remember. So prioritizing where it needs to be prioritized.
Okay. And I suppose then figuring out as well, because we've all got too much to do, right? We've all
Yeah,
got
always.
lists of things that we're never going to get to the end of. So I suppose you talk about prioritizing there. It's also about understanding, well, which of these things is important and which are the things that are going to make the difference.
Yeah, and it's planning them in the calendar. It's giving
Yeah.
that. But, you can do it the same. The science versus the one-to-one with the people you're line managing and those things. You can tweak it over sort of weeks, but months. You sort of realise then you're losing touch on some of the projects. So I think it's a balancing act, and then using the electronic things. So I went to an iPad quite quickly, to take my notes, and they've been electronic. So now my OneNote pieces, 2 minutes before I call into it, I do the search like I did before I called in, and I had all my notes from what I spoke to Emma. I was like, oh, okay, I've got that. I've got the agenda. I'm ready to go. And they're on my phone now. So if I'm in a networking and I bump into someone, I think, oh, I know them. It's just, okay, oh, yes, we were talking about such disease. So I think there's a lot of electronic pieces as well that help. I can see my home calendar at work. So when we're sort of planning, I put it in both. So
yeah, I think that's a really important point, tied to the hyper-focus stuff you were talking about, is that if you are going to be engaged and present in whatever you're doing, you have to have ways to quickly pull the information that you need so that you're not spending the end of the last thing preparing for the next thing, right?
Yes. And it's great. So I've heard lots of things, when you have 45-minute meetings. So particularly when you're in big places like AZ, it could be a 5-minute walk to the next, to where you're having your next meeting.
Yeah, true.
But it's a little bit of that regrouping and getting ready for the next one. And then I'm surrounded by a very good team here that have that information as well. So I've got Kishan in the room here that accompanies me a lot of the time and has a nice big database where we can quickly call up information.
Fantastic. So making the most of the tools that are there for you.
Tools and people, yeah. I think it's finding the synergy where and it doesn't always work. The biggest headache in the moment is my dog, because my children can look after themselves, but the dog can't be left for 8 hours now that we never know who's in and who's not. So
fair enough. And the dog's not so hot on the calendar and
no, he's not very good at that at all.
Yeah, fair enough. And I mean, along with the things you've already talked about, any last things that you'd like to leave people with, or any pieces of advice that have been particularly impactful for you?
I think it is about having the right mentors,
Okay.
finding those people. That's a good thing. And personally, I just value networking.
Right.
Just those the connections from a long time ago and building those. But I've spent 20 years building up my oncology network, and then I've come into a job where I need genomics and investors. So I'm almost back to okay, how do I get out there? How do I meet people? And that's one of my learning styles. I've done enough of the Myers-Briggs and everything else that drip feeding of information is the way then that I sort of absorb and then go, oh, okay, we probably need to go in this direction because, I've heard it from 5 people. This is a good, these are good people to talk to, or that's going to answer this question I've got over here. But people starting out, I would say, yes, networking is key and developing, because you never know, what your projects might change, the science might change, or, you were thinking about different countries. I recently went over to China with the HSBC and, touched base with one of the chief of staff that I'd never met over there. Right. We didn't actually meet face to face, but we did manage a good email chat. So those connections are really important.
Yeah, absolutely. And I suppose to wrap up, you told us a little bit about where things are at Enhanc3d Genomics. What would you like people to know about what's next?
I think it's the speed that we hope to move at, the ambition that we've now got. It's taken me less than 6 months to fully believe that this could be that, what would you call it, the next dimension in discovery.
Right.
And we've been so many people are using genomics, but it doesn't feel like we've had that sort of health breakthrough that it's always promised. So really, the next step is to consolidate that with the right collaborations with the pharma and biotech partners to show and demonstrate what it can do, and then hopefully to scale up at a speed that doesn't disrupt us, but really informs the research community.
Yeah, fantastic. Well, exciting times, and we wish you the best of luck.
Thank you very much.
Thank you so much for joining us.
It's been a pleasure.
Thanks for listening. If you enjoyed the episode, please like and subscribe and share it with someone else you think will benefit. And for more podcasts, videos, blogs and insights, come follow me, Tom Froggatt, on LinkedIn, YouTube, Instagram, Medium or TikTok. See you next time.